Susie, no, wait, Emily.
Rob.
Emily, how much do you know about Ediacaran evolution?
So I know a fair amount about Ediacaran evolution because it's what I spend my time thinking about.
Yeah, indeed. That's a rather contrived question, seeing as you spent your entire academic career working on that. So, just to explain to everybody, Emily is our guest today to pick up where we left off with a discussion with Susie last time, talking about Ediacaran evolution and the story of sex. Susie, are you ready? No, it's Emily. I'm getting confused already about who's who. Emily, are you ready?
I am ready, Rob.
Okay, so Dr. Emily Mitchell is at the University of Cambridge, and you've been there for what, 10 years? No, more than that, 20 years. How long have you been at Cambridge, Emily?
Since 2008, but I started off in earth sciences, but I've been in the zoology department since 2019.
So you and I have known each other for a long time.
We have, yeah.
We've talked about all sorts of things, hanging around. We've shared a lot of parenting tips, I think, in the most part,
We have
and the life of being an academic with kids, and just generally particularly during COVID, yes. But so it was all the bizarreness as to why I didn't invite you on to be the guest when we were actually talking about your paper. In retrospect, was a mistake. So I thought we should have you on to correct the record and also ask you more broadly about it. So question number one, Ediacaran or Ediacaran? Susie and I could never agree.
Ediacaran because it's named after a place in South Australia, and that's how the local people, the Aboriginals, would pronounce it.
So is there a particular group of Aboriginals in that part of Australia, or are they like a multitude of peoples?
There are a few different groups.
Okay, so Ediacaran, is that right? Okay, I'm going to follow that from now on. So, for those of you that don't know, the Ediacaran is a sort of weird, esoteric kind of part of the fossil record, but it's also really important and fun. But it's a real challenge to get into. So, why do you dedicate so much of your time and effort to work on the?
So there are two reasons. One is that it's where we first see animal fossils appear in the rock record. So if we want to understand the origins of animals and how they first evolved, you want to be working in the Ediacaran. And the second reason that I enjoy working there so much is you have absolutely amazing data. So if you think of the fossil record, it's normally stuff with bones or shells. And so it accumulates over very large time periods. Whereas Ediacaran, you have entire communities that were killed in an instant where they were living. So you have census populations. And so when you're thinking about how to understand these fossils, you can use a whole host of techniques that you normally aren't really very appropriate to the fossil record.
Yeah, it does seem really hard to work on that. I mean, it's also, I guess I was also asking because that's a really good explanation about why it's important. But it's also, I guess I was thinking about as a little girl, do little girls grow up dreaming, thinking of, I'm going to work on these weird fossil surfaces? I mean, presumably that's not how you came to it. Although Tina Negus did. We talked about Tina Negus last time.
Yes. Oh, yes. So yeah, Tina's wonderful. She does amazing artwork of Charnwood and the fossil surfaces. So no, as a little girl, I was interested in sharks and maths, and I ended up doing a lot of maths at university before going back to more biological sciences. And my PhD originally was supposed to be on the Burgess Shale, but I got a bit stuck because the preservation biases, how do you incorporate them if you're trying to think about the kind of quantitative community structure? Whereas in the Ediacaran, the preservation biases are much, much, much less. You don't have to worry about time averaging. You don't have to worry about stuff swimming around and whether or not it's captured. So
Yeah,
it's a lot easier in that respect.
I find that confusing that you say it's easier to work on the Ediacaran rather than the Cambrian. But maybe that's because you're a maths bent. So you've been into maths for a long time. And indeed, that was one of the problems we had when talking about your paper was that maths is a theme that goes through all of your research, right? Big hardcore maths to think about paleoecology. But you didn't get really trained. I mean, you were trained as a biologist or geologist. How do you get all the maths training, or is that just something you have to do independently on your
No,
PhD?
well, so my undergrad was in theoretical physics. I did a master's in math.
Yeah, of course.
So actually, my training is in maths and physics. And I did a master's in biology and in ecology. So it's only during my PhD that I started learning earth sciences.
So you're late to the story. All right, fair enough. We've just covered a whole episode coming up on how to become a paleontologist, so we'll have to include the theoretical physics as a entry-level pathway into that at a later stage. So I want to know a bit about the work that you do out there. So you've done lots of maths, but you're also out there busy collecting the data, right? So I've seen pictures of you looking very dynamic and exciting out on the Newfoundland shoreline. What's it actually like to work out there?
So I mean, it's really lovely. We're heading out there on Saturday actually, starting a new grant. But I first went there in 2010, and for the last 10 years now, we've been laser scanning the fossil surfaces. And so it's absolutely amazing because you've got all this wonderful coastline and all these outcrops covered in fossils. And sometimes you just get one or two Ediacaran fossils, and sometimes you get literally thousands. It depends on how much outcrop you've got. Um, in terms of what it's like working there. So the laser scanner we use, you need to have good weather. So it's very weather dependent. So there's a lot of checking your weather apps, trying to predict where the rain's going to come in, if it's going to come in. But then if the weather's good, you go out to a surface and you'll spend the day sitting on the rock surface laser scanning.
So I imagine it's quite isolated down there. I don't think a lot of people live around there, but maybe I could be wrong. But also, I now got this idea of you just sitting indoors having cups of tea, waiting for the rain to go away. This is this is my kind of field work now. I can about get my head around.
There is some of that, watching the rain, hoping for it to disappear. So, I mean, in Newfoundland, there are kind of three main localities where we work. And Mistaken Point is a very famous one. It's UNESCO World Heritage Site. It's got the E-surface. And that, you've got Portugal Coast South is nearest town. There's about 150 odd people who live there. And then you're looking at maybe half an hour drive to the parking spots where the tourists park. And then depending on where you're going, 45 minutes to an hour and a half to the fossil side. So it depends. I grew up central London, so it's very isolated for me. If you grew up in like Canada or the States and it's, or Australia, then it's relatively unisolated, right? You're still 2 hours drive from a hospital. But then around in the Bonavista Peninsula, we stay in Port Union, and that's absolutely amazing because that's very different because it's very, very built up. So I gave a talk at a school there once, and they were teenagers. They were wonderful because they couldn't quite comprehend that we'd come all the way from England to look at their fossils.
It's interesting. I mean, do they? I thought they might. I thought it would be big news in Newfoundland that hey, we sit in one of the most important geological places in the world. Is it not something they know about?
So I mean, it's things have changed a lot in the last 10 years, but just behind the back of the school, there are actually fossil sites. So, literally around Port Union, Little Catalina, there are some times where you have people who historically have been putting their boats out over fossil surfaces because they didn't realize, and then obviously things are very different now, but you literally are just walking around and they're kind of very much enmeshed in people's lives. And so when we're walking out in around Port Union on the Bonavista Peninsula, you get people berry picking for a lot, for example, or going for hikes. And so there are it's relatively busy. And there you're maybe looking at a half an hour walk or so, 45-minute walk. It's not far. But yeah, there are lots of lovely berries in the at the right time of year.
I saw an interesting talk from Jack Matthews who's talking about the site in Leicester, whereas actually trying to protect the site is difficult because the more attention you bring to it, maybe the more people might be unwanted. And maybe the best thing to do is to keep it unknown. I don't know how that works in Canada, though, because there's a very different balance of.
So it was, so I've kind of been part of the China Research Group, as it was then called, for decades now. And this was one of the big discussions we used to have, because if we publicize them, then people will know about them and people might steal them. But in reality, in Charnwood, you had teenagers drinking and graffitiing actually on the fossil surfaces. We assume they're teenagers. And so there was actual real damage from people who didn't know versus kind of the concerns about potential fossil theft. And so that's been a kind of discussion that's been ongoing for quite a long time now. And so I think in Newfoundland, it is a lot more, again, in Mistaken Point is a lot more isolated. And there was a potential theft a decade or two ago. Some creationists tried to steal a fossil because they thought it would prove that God exists. But what was quite funny is you've got all these beautiful Ediacaran fronds, and they tried to steal the Ibs, which is like a blobby mess. It's like a microbial colony that grows up around the carcass. So it's like of all the fossils to try and steal, but the locals managed to kind of, scare them off.
That's nice. Thinking about the creationists and the misguided attempt to try and do that. That reminded me about when I had a paper come out a long time ago that was in one of my biggest papers was about looking at preservation modes and how actually they can distort fossils and make them look a bit more can distort the evolutionary inferences that you draw from them. And I remember trying to follow up on the press coverage, and the creationists had sort of covered it and said, "Oh, this is rubbish because none of this doesn't mean anything. This can't help you understand the fossil record." And I found that kind of funny because if they'd actually read the paper, they could have weaponized that for their advantages. They could have turned around and said, "Look, according to Sansom et al., these fossils are distorted and the evolutionary tree is wrong." But no, they just dismissed it out of hand. I thought, read it. You could, if you wanted to, use that. Now, obviously, that wasn't the point of the paper. I wasn't saying that. Anyway, I guess anyway, we shouldn't go on a creationist rant here, but that's interesting that the fossils gets damaged. So one of the reasons that the scanning surfaces work so well is because these fossils are like living and dying in the same place, right? So you've got this lovely pristine surface which allows you to map them as they lived and died, and therefore do all the interesting ecology, which is what we talked about in that paper, where you were able. This is going to be awkward as I now explain your own paper back to you, but as a reminder to everybody, Susie and I covered Emily's paper last week, where we looked at how the clustering and the distribution of those fossils on the surface was indicative of different types of reproduction and the competition, and that there was a relationship between the types of sexual and asexual reproduction and the level of competition between and within species. So, question for you, Emily: what did we get wrong? Did you have a chance to listen to that episode?
So we talk very much about asexual reproduction, which is using the filaments like spider plants or stolon. And in terms of the waterborne current reproduction, it is most likely to be sexual reproduction, but we actually don't know whether it is or not. So I think in modern benthic animals, about 86-90% of waterborne propules are the result of sexual reproduction, but not all of them. So in theory, they could be tiny, tiny little fragments or buds. It's unlikely, but can't necessarily rule it out.
So we made a major error there. Do we need a scolding?
Well,
You could say yes. You could say yes.
it's not a major error. I think lightly sexual reproduction is the phrase we use in the paper.
Okay. So that's with the drifting off into the ocean. So I guess, what do you think we missed about your paper? What do you think? How would you describe the major implications for this?
So if we think about things in a very naive way, you get animals originating and they've kind of got wide open ecological space. Like they don't have predators, they don't have to compete with anything. So they could have radiated really, really quickly and filled all available niche space. But if you look at the published records of diversity, you don't see that many new species coming along kind of from about 574 when we first see them till kind of 555 when you're in kind of Australia and the Flinders in the White Sea region in Russia. Then you see a massive radiation. And so kind of I was always kind of curious, why do we see this very, very slow diversification? And I think what's going on and what we showed is that because you've got this kind of reduced competition and reduced selection pressure, that there's a lot of weak competitors that can survive. So they're not actually being forced to innovate.
Cool. So at the risk of mansplaining your own paper back to you again, this was the topic that Susie and I talked about, with the idea that when these Ediacaran animals, or proto-animals, if we want to call them that, Ediacaran, sorry, I'm going to have to... When these Ediacaran proto-animals turn up, there was a long period of time, about 10 million years, where they're not doing an awful lot. They're being quite simple until later on when they start diversifying. Right. And then your inference was that that's because of the different amount of competition and reproduction going on in those two phases. So I guess one of the things we touched on very lightly was that we have this sort of classic idea of the Ediacaran, where we've got this sort of slow, simple Avalonian thing for about 10 million years. I'm plucking these numbers off the top of my head. And then we've got the more complex White Sea things that turn up later on. And that's kind of where you planted your study. But that paper came out from Scott Evans and crew recently, where they found this more complex things in Canada. So they found was it new? I don't know if it was new, but anyway, they found this deposit in northwest Canada that had Dicksonia, Kimberella, Io Andromeda. So these complex, more complex things that might be moving about, but they found them earlier in the Avalonian. So does this mean a mix? No, you're shaking it.
Well, kind of to some extent. So this is a really, really interesting study. So everyone was aware of these fossil sites, but the fossils had only been found in float. And so what Scott and his team did is they went and they actually found the surfaces, and then they went through. And what's really, really special about this site is it's got two things which hasn't been found before. It's got a long time range, so from kind of over 567 to 538, but also ranges of environments. So it goes from deep to shallow.
Yes.
And so one of the big questions that's been going on for over 20 years now is we've got the kind of classic assemblages. So these are where different fossil sites are grouped based on similarity of the species that inhabit them. We've got the Avalon, the White Sea, and the Nama, and they all, to a first approximation, occupy a different environment and different time. Now, 20 years later, we've got a lot more overlap. Assemblages like the Shumantan in China, which has got some White Sea, some Nama-style fossils and overlaps a lot in time. But the big question is what we're seeing directly in the fossil record, this kind of deep to shallow water radiation, is that an evolutionary trend, or is that just a result of kind of a lot of biases in what environments we're preserving fossils in?
Yeah, absolutely. That was what the point Susie and I talked about last time, was what we started trying to talk about, how it was really hard to disentangle the ecology from the environment. We talk about changing things through time, then we've got these patterns of preservation of the different environments. And I guess this paper sort of breaks that down a little bit, but still we've got these big biases all over the place about deep sea versus shallow water. Although, interesting side note, which is just an opportunity for me to start talking about my own research again. This is becoming into a theme this episode. Was that in that paper they started talking about how they've got this, even they were inferring a sort of potential pattern from deep sea to shallow sea as things originating in the deep sea and then moving off sea. But actually that goes in contrast to a lot of papers, including one of my own, which with a lot of co-authors, for the most of the time, is that things generally, marine life seems to generally diversify in shallow environments and then spread out to rivers and spread out to deep sea. But the picture from the Ediacaran is the opposite. I mean, what do you think?
Well, so I think that's really, really interesting. I think this paper goes a long way. So part of the issue, so what this paper says is that it's got all these different environments, and what you see is your oldest is deep water. You don't have shallow water fossils. Then as you get younger, you start to see some of the White Sea fossils coming in the deep water. So deep water Dickinsonia, which is just totally crazy for people who've been here doing it as well. But then as you get into the proper kind of White Sea time zones, that's, kind of, I guess, 550 to 560, you're seeing a lot of fossils in the shallow waters and through all the different environments. You're seeing this transition across the different environments and across the different depths and times of this transition from Avalon to White Sea to Nama. So what this paper implies to me is that the differences between the assemblages aren't just an environmental thing, but you have this missing data, right, from the shallow water older stuff. And this is a perpetual problem, like, because we don't have any kind of older than 5, 6, 7 shallow water animals and what's going on. And so and this is counterintuitive, as you say, because life is generally more diverse in the shallow waters. You have lots of diversity patterns come from the shallow water and into the deep. Well, so part of the issue is that a lot of people over decades and decades have tried to find shallow water old Ediacaran fossils, so 565, say, to 580, and they've gone and spent weeks and weeks in the field and not found anything, and then they don't publish because you don't publish a paper saying I just spent 3 weeks not finding things. Yeah. And so at what point do you say, well, we know enough to know that's not there? We also, at older shallow water, we get microbial mats, we get algae, we get protists, but we're not getting animals. So of course there could be a taphonomic bias, but what sort of bias would you get microbial mats and not even discs?
That raises a really interesting question, because in paleontology we often talk about biases in terms of, well, we've got this preserved and hopefully we'll find this some other point later on. If we look for dinosaurs earlier on, maybe we'll find them if we look in the right kind of deposits. But the other interesting ones is with mammals or reptiles or all sorts of things. These sorts of predictions are made. But in this case, what you're describing is potentially hard absences, in the sense that we do have those shallow water deposits, but they're empty of animals, which means that rather than it just being a not described, it might actually be a hard absence, which is interesting.
Yeah, and I've got a lot of colleagues who disagree. A lot of colleagues will say, well, it's not exactly the same facies. And it's like, okay, sure. But also, there is also still a lot of people that haven't published on potentially more similar facies. But also, I think you need some special pleading if you're getting microbial mats and you're getting algae and you're not getting animals. It's like, could there be a pathway? Sure, but why wouldn't you see those remnants of those Ediacaran fronds? What's going on? They're living amongst microbial mats. and you're seeing the map.
Why not these
Seeing
mats?
the maps.
Hmm. Great. So I should have mentioned the paper was Discovery of White Sea Assemblage Fossils from Laurentia, which was published in Science Advances in May 2026 by Scott Evans. We mentioned it last time. But the other paper I wanted to ask Susie about, not Susie, again, I'm making the mistake. I'm completely stuck in the default of talking to Susie. Susie, this is not Susie, this is Emily. Goodness gracious, Emily Mitchell is the author here. This week, the other paper I wanted to ask Emily, as our resident expert on Ediacaran, was this other paper that just came out in between Susie and I recording and then Emily and I meeting, which was earliest evidence of behavioral handedness in the Ediacaran motile bilaterian Spragyna, which was published in Scientific Reports in July. So this is like fresh, hot off the press. So, Emily, how would you describe Spragyna to people that have never heard of it before?
So people often find it one of the more exciting Ediacaran fossils. I think it is the state fossil for South Australia. It looks like it's got a little head plate, and then it's got all these little segments that it kind of goes down the body. So originally people were wondering if it's potentially an Ediacaran arthropod or some sort of segmented worm. The best way to wind up Cambrian arthropod workers is to talk about Spragyna being an arthropod. They hate it. The segmentation is very
It's got
different.
no legs, surely. Well, I guess some arthropods don't have legs, but you can't have your first arthropod with no legs. That would be upsetting.
Yeah, and the patterning, the segmentation. So it's really interesting. It's in that it's bilaterian for sure. It's got this complex kind of posterior anterior differentiation, which is not all that common, I guess, in Ediacaran.
Sure. I might call it then a segmented lozenge with a front, because it has one end. Whether it's a head or not, I don't know. It doesn't have any organs that we can see, right? It's just a segmented sausage shape with one element which has a sort of, well, I can't think of another word other than front. What would you call it?
Anterior region.
Anterior region. Let's not call it a head, because that's a loaded question about what is and isn't a head. A cathalization is a long display. Anyway, so let's have a look at what these guys did. They went and this is Scott Evans again. This is another Scott Evans paper with a bunch of people from Australia and around. They went and looked at the surfaces in Australia and tried to count these guys up. So they found 75 Spragyna, and they found that 70% of those are bent. So again, if we're thinking about this as a lozenge or a worm, they're a bit sort of more bean-shaped, I guess, or banana shaped, depending on how you want to call it. And I guess the bit that is the weird, this isn't really a massively important paper, but I guess I just find it really interesting because it's just a weird, quirky one, right? They found that of those ones that are bending, majority of them, significant majority of them, were bending to the left rather than to the right. And therefore they infer that this might be behavioral and that Spragyna might have been moving. So do you think Spragyna is moving?
So I think it's quite likely. There's no evidence of trace fossils that it can move, but it does seem kind of just from its morphology that it was something that could move around. And I mean, I really like this paper as well. I think this makes a lot of sense.
That's boring. I want you to, it's more fun and dramatic if you can come in and pour scorn on papers than if you're coming in and praising them.
You picked the wrong papers.
Well, hang on, I've got one in the bank yet. Don't worry. That you may be able to pour scorn on. I should have explained why this is so important about whether it does or doesn't move. I mean, if it might feel like an esoteric question about why this lozenge-shaped Ediacaran fossil is or isn't moving around, but this makes a big deal about it, your interpretation of it being what sort of animal it may or may not be. Because if you're moving, then you're an animal. I mean, that's an oversimplification, but let's be clear about this. And if you're moving in a particular wormy type way, then you're a more advanced kind of bilaterian type animal. Sorry, I cut you off when you were praising this paper, and I was busy being rude. What nice things were you going to say?
So I just say it's very, very interesting that they do have this left-handedness. I think there was a study recently out on humans that said that people similarly always trend towards the left if they're walking. That if left to their own devices, people will slightly bend over to the left. So that is quite interesting. And I think they did a really good job of talking through why they didn't think it could be a taphonomic thing. Because you can imagine if everything gets swept along a bit, it could try to kind of compensate for that. But I think they did a very good job.
Well, yeah, I've got a quote here. They said post-mortem muscular contractions, well, we don't know if it has any muscles or not, or desiccation would be expected to impact all specimens of the same fossil surface equally, thus rejected here. I wasn't quite so sure about that. I mean, I've rotted a lot of things. They can move in quite weird ways and end up looking bending, especially when we did that with worms. But we'll allow it. We'll allow it. You're happy with that. And if you're happy with that, I'm happy with that. So let's move on to another quote from them, because this is really gets into the crux of the matter, I guess. Quote, based on neuronal capabilities of extant taxa in which this type of behavior is recognized, we contend that handedness is suggestive of relatively advanced nervous system in Spragina. Now, I need to play the little jingle for Speculation Station here, because let's get into speculation.
Speculation Station.
What do we think about that claim?
If something's always turning left, does it then demonstrate a superior ability to think about things? I mean, it shows that it must have some relatively complex neurology going on in its head, I would have thought. Is it more complex than a sponge? Probably.
Well, I guess this feeds in, this piqued my interest, but I guess because I was thinking about this, because we talked about on the fossil files, we talked about lateralized behavior before, and I guess first of all, with whether or not we don't even know if Spragina had a nervous system, right? There's no reason to think that it would have done. I'm not entirely convinced that turning left or right would indicate a complex nervous system. They cite a paper, but the one that we talked about most recently was the giant octopus story from Japan. I don't know if you saw this, but it was in Science. They studied got these massive octopus beaks from the Cretaceous of Japan, and the inference from that paper was that because they had lateralized behavior, that one side of the beak was much more worn than the other side of the beak, and they inferred that therefore it was a super intelligent predator because the behavior was going on one side than the other. And I guess this is picking up on that theme, is that this is a handedness kind of behavior and implies that it's complex. I'm not entirely sure I see the biological rationale for handedness being necessarily a complex, intelligent behavior, and I don't think that Spragina can be as intelligent as a giant octopus.
Well, who is as intelligent as a giant octopus?
Yes.
I mean, if you think of those deep sea fish that have kind of one eye much bigger than the other, because they always kind of are on the side and looking up, they will have a handedness, but that's kind of more an adaptation to needing one side more than the other. So I think that if you've got a handedness doesn't necessarily suggest that you're thinking about what you're doing, which I think is the implication. Like with the octopus, if you're eating something from one side all the time, it's constantly. But again, if you have an asymmetric morphology, you might be doing it just out of function, I guess.
Well, that's the same question about Spagana, right? So a lot of these Ediacarans, goodness gracious, it's going to take me a while. Ediacarans often aren't necessarily symmetrical with left-hand side and the right-hand side. They've got a slight offset, right? So what about Spagina? Do we think that has a left-hand side, right-hand side offset? Or you're looking at me to suggest that I'm working off old information and that has been debunked, that there isn't offset symmetry anymore?
So, yeah, you have what we call glide symmetry quite a lot in the Ediacaran. The Spragina is pretty bilaterally symmetric.
Okay.
So Dickinsonia, a very famous one, is sometimes referred to as having glide symmetry, which it does in some locations, but in others it doesn't. It's wonderfully, perfectly bilaterally symmetric.
I didn't know that. I'll have to update my teaching materials where I talk about Dickinsonia a lot. We should have explained what Dickinsonia is. It's like a big flat pillow, I guess, that just sort of sits on the surface. A pancake is better than a pillow. A pillow has much more dimension than that. It's a very cheap pillow, if that's it. So it's very, very, it's very flat, and it just sits on the surface, digesting and then floating off and then digesting again. Anyway, you were saying about Spragina, so they are more symmetrical than I first realized then.
Yeah. So I mean, they look pretty bilaterian to me. I think you'd need quite a lot of special feeding to say that they weren't bilaterians.
Okay. All right. Okay. Cool. I'll try and have to think about more antagonistic questions to get you going then. Well, here's one for you. Final Ediacaran question for you. Seeing as you're Ediacaran expert in the chair today, I can quiz you on lots of Ediacaran things. One, we've covered a few Ediacaran episodes. I guess one was the most recent one where we covered your own paper. Thank you for covering that. One was we looked at the sort of merging and blurring between the Burgess-Shale and Ediacaran boundaries. We talked about that recent paper from China.
Yeah,
that's fun. And the one we did way back again was part of our Fossil Fails episode, where we looked at a series of paleontology gone wrong. And one of them was the Ediacaran beehive found in India. So just as a reminder for listeners, if they're not aware of this, this is the... I brought this to the attention of Susie, who had never heard about this. This was a supposed discovery of a really interesting Dickinsonia-like fossil in the Ediacaran hills caves of India. And then the discovery about a year, 2 years later, that actually it wasn't a Dickinsonia, it was a whole load of beehives. Modern bees had just left behind a waxy remains. Susie and I had a good laugh about this. Susie was flabbergasted about how anyone could make that mistake. we had a conversation about pareidolia and image searching. So I was trying to be forgiving and thinking, well, if you're searching for the pattern for a Dickinsonian like animal all the time, and then you see something that looks like that, I was trying to give them some forgiveness. How did that happen, and what do you think?
So, I mean, you're absolutely right. We look for patterns and things. If you spend too much time laser scanning fossils, you go back and start seeing them in the wallpaper. So I think there's some wonderful researchers on that paper. There's also one of the authors is quite commonly finds patterns where other people don't.
Are we talking about Greg Resalak here?
Yes, yeah.
We did describe him as not a maverick, a crank, I guess. He is at odds with a lot of the other researchers in the field, right?
Yes, yeah. Yeah, so he, for example, thinks that Ediacarans are all lived on land and are lichens and lichen-like organisms, and he doesn't seem to take on board anyone's, evidence to the contrary. And so, yeah, if he sees something he thinks is Dickinsonia, he strikes me as the sort of person who'd take the best possible photo to make it look, most like a Dickinsonia, less like a beehive.
I knew if I get you there eventually, you'd slam somebody. That's not entirely true. I'm being unfair to Emily here. If you want to find out more about that episode there, but I guess this raises questions about how you might do the fieldwork, because it wasn't even on the bedding plane, right? It was just a flaky, waxy surface that came off in the hands of people.
Yeah.
I mean, I take it you've not been to this cave in India to go and have a look yourself. Well, you would definitely not need to, would you? Because it's not. You're not going to.
Exactly, it's not. Although I think there are some really, really fantastic Indian Ediacaran researchers, and they're finding some really fantastic stuff. In this case, it was not a Dickinsonia, but.
No. Yeah. So this is, I guess, the question. That's a neat segue onto the final question I was going to ask you. What do you think's next for Ediacaran research? Where will the big discoveries come? Where should we be looking?
So I think what's really, really key is more field work. So as Scott's demonstrated, even known localities can still come up with really, really new and novel insights. There's still a lot of surfaces that haven't been published or have only barely been touched on in Newfoundland. But there's also got a lot of colleagues doing field work in Australia and Namibia, for example, or Morocco in Ediacaran rocks, finding new things. So I think that's really key, is going out to more Ediacaran localities and having a look for more fossils.
Lovely. So I guess that raises a lot of questions about the search pattern again. So Ediacaran fossils are not necessarily very easy to recognize, right? I used to live in Leicester, so I've been to the Bradgate Park to go and have a look around and tried to stroll up and down the surfaces and have a look and find them myself. As a professional qualified paleontologist, I should point out, who knows exactly what he's looking for and still can find nothing. I mean, maybe this is speaks to my absolute lack of skill as a field paleontologist. But I have spoken to some people subsequently that actually they're quite difficult to find unless you know specifically what you're looking for, and you need to go at a certain time of day where the light is in the right place. But I guess this is a difference from, say, research in the Devonian, where I might kick over a rock and see a nice bony fish or a piece of wood or whatever. Well, not wood, but it's Devonian. Well, I might find I don't know, a trilobite or whatever. And it's immediately recognizable as something biomineralized and with a distinct shape. Whereas if you're looking in the Ediacaran, you've got to be a specialized person, right? If you're looking around these flat surfaces, some of them look like nothing more than a rough blob that it could easily be overlooked by lots of people in the past.
Oh, yeah.
Yeah.
Indeed. Yes.
That was a long question. Yes.
Okay. Yeah, they could. I mean, so in South Australia, some of the Aboriginal paintings, I can't remember how old they are, have things that look a lot like the fossils.
Really?
Yeah. And a lot of the locals around Newfoundland will talk about fish bones in the fossil sites. Oh, well, fish bones in the rocks, which look a lot like fractifusus, for example. So I think there is very much about kind of getting your eye in. I think if you sent out a vertebrate paleontologist to try and find them, they'd probably be less likely to find things.
So now you slam vertebrate paleontologists. That's great. Good.
I think it's fair to say that vertebrate paleontologists are probably less likely to find Ediacaran fossils than Ediacaran paleontologists.
Yeah, that's not already a slam. That's just a basic statement of fact. That's okay.
But yeah, so you need very experienced field geologists so they can target the right sorts of rocks. You need people who are experienced at looking and finding Ediacaran fossils.
I was also going to ask you where in the world we should be doing this, but you already said that. You think Morocco and Australia. Morocco's an interesting one because we've covered Morocco a few times because of the big uptick in. There's all sorts of discoveries coming out of Morocco at the moment, from the Jurassic, the Cretaceous, the Ordovician, but I hadn't really heard about much about Ediacaran going on in Morocco.
So there's been some discoidal fossils and obsidiomorphs found there.
Oh, nice.
So so it's potentially very promising, but we haven't got the mistaken point style.
So for one of our special bonus episodes, Susie gave a longer talk about her trying to set up more, helping the local communities in Morocco build more paleontological infrastructure like museums and UNESCO world sites and things like that. So they're having a bit of a moment there with the sort of budding of what's going on in there, but I didn't realize it was such a rich geological range of stuff going on there. That's quite exciting.
Yes.
Yeah. Oh, well, thank you, Emily, for joining us. Normally I ask what the take-home messages are, but I guess this is a bit weird for you because your take-home messages are your own research. But let's do that. What do you think the take-home messages are? What would you like people to go away remembering or knowing from all the different Ediacaran topics we talked about?
I think the key thing is that Ediacarans are the start of animal life. So if you want to understand if we want to understand animals, we should be thinking about Ediacarans.
Oh, I'd heard they were all just lichens.
You know how to wind me up.
Okay. Just for the sake of the record, they're not lichens. This is okay. All right. Okay. Well, thank you very much, Emily. Normal service will resume next time with Susie, but I really appreciate you coming along and sharing the time to chat with me about all things Ediacaran. Thanks, Emily.
The Fossil Files was created by Robert Sansom and Susanna Maidman.
Fossil Files is a labour of love created in our spare time with no budget, so we really welcome any support you can provide us via our Patreon.
There you can access bonus content and join in the discussion by posting your own questions or even participate in live episodes.
In either case, we want to hear what you think. You can let us know what you thought about the topics we discussed or make suggestions for future papers in our social media channels.
You can find links to our Blue Sky, Instagram and YouTube channels on our website fossils.libsyn.com.